Welcome to the first lecture in Unit 1, Chapter 1, the main themes of microbiology. To start, we'll group all microorganisms into 5 types. Microorganisms can be referred to as microbes for short, and generally they are all organisms that must be seen with a microscope, but we'll see with the first group, the helminths, that is not a requirement. The helminths are parasitic worms. They are multicellular, meaning they are made up of many cells coordinated together as a single organism. Those cells are eukaryotic and have a nucleus, and they are considered macroscopic because you do not need a microscope to view an adult helminth. They are still considered microbes because points of their life cycle are microscopic, and you must have a microscope in order to see those stages of the life cycle. The second type of microorganism are the fungi. These are mostly decomposers and break down dead tissue. They are uni or multicellular. Their cells are eukaryotic and have a nucleus, and they also have a cell wall. The third group are the protozoa, which are unicellular organisms, meaning made up of only one cell, and that cell is eukaryotic and has a nucleus. The fourth group are the bacteria, which we will largely focus on in this course. The bacteria are unicellular, so one cell is an individual organism. That cell is prokaryotic, so does not have a nucleus, but it does have a cell wall. The last group are the viruses. Viruses are parasitic. They live inside another host cell, and they themselves are not made of cells, so they are acellular. Viruses have DNA or RNA, which, unlike all other life forms that are made of cells, these other life forms have DNA and RNA. So viruses are unique in that they are not made of cells and that they don't have both DNA and RNA, but have one or the other. Of all the types of microorganisms, the viruses are the smallest. All life forms can be organized into 3 domains, which are the largest grouping in the organization of life. The first domain are the bacteria, and the microorganisms of the same name fall into this domain. So all bacterial microbes are in the domain bacteria, made up of unicellular organisms that are prokaryotic. All other types of microorganisms except the viruses are in the domain eukarya. This group of living things is made of cells having a nucleus, meaning the cells are eukaryotic. In the domain eukarya are the fungi, the protozoa, and the helminths, which would be considered animals. Humans are also considered animals and are in the domain eukarya because our cells are eukaryotic. There is not a categorization of viruses on the phylogenetic tree of life because viruses are not made of cells and are technically not considered living. In naming a microorganism, we use the genus and the species of each microorganism. A genus is a group that is more specific than the domain in the last slide of the phylogenetic tree, but still contains several types of species, where each organism is a unique species. When writing out the scientific name of a microorganism, we capitalize the first letter in the genus, and we use lowercase for all of the letters in the species. The genus and species names are also written in italics and can be abbreviated in this example of Lactobacillus rhamnosus as L. rhamnosus. Microbes affect humans in many ways. We will start by discussing the beneficial effects of microorganisms on humans. And largely, microorganisms are beneficial to humans. One example of how microbes benefit humans is by conducting photosynthesis. Microorganisms that can do photosynthesis are able to make food from sunlight and release oxygen in the process, which results in the atmosphere of the earth containing oxygen that we breathe. Microorganisms like the fungi are responsible for decomposition and nutrient recycling. We use many microorganisms for environmental cleanup, which is a vocabulary word shown in red, bioremediation. Bioremediation is the use of microorganisms to clean up polluted areas of the earth. The microorganism can break down chemicals into less harmful molecules and make the site less polluted. Biotechnology is the use of microorganisms to produce industrial products like foods and medicines. In humans, we have a microbiome comprised of viruses, fungi, and bacteria that keep us healthy and are present in a normal, healthy human. We also use microorganisms for food production, like dairy products, and we use them for producing alcohol, like beer. The negative effects of microorganisms on our lives is that they cause disease. A microorganism that causes a disease is called a pathogen. In this listing, we have the highest causes of death worldwide in low-income and high-income countries, and several of these diseases are caused by microorganisms. The history of microbiology will begin with Anton van Leeuwenhoek, who was a Dutch scientist that was good at designing lenses for magnification. Shown in this image is van Leeuwenhoek's magnifying glass, which was the best in its time and could magnify cells at a greater factor than any previous microscope. Because of the power of his microscope, van Leeuwenhoek was the first person to observe a living cell. This discovery of the cell led us to the conclusion that all life is based on cells, and opened our eyes to an invisible world that was now made visible. This will have huge implications for understanding the cause of infectious disease. Joseph Lister, shown in the image on the left, was a physician responsible for developing sterilization and aseptic technique, which is attempting to reduce the number of microbes in a medical setting. Shown in this image is the use of phenol vapor during a surgery procedure to kill microbes and prevent them from entering the patient's wound to help the patient heal and survive the surgery. Additionally, Alexander Fleming discovered antimicrobials, which had a huge effect on reducing the rate of infection. As shown in this data, around 1920, the maternal mortality rate in giving birth dropped dramatically around 1920 when Alexander Fleming discovered antimicrobials that could be used to prevent infection caused by childbirth. Another player in the history of microbiology is Robert Koch, who came up with Koch's postulates that linked microorganisms to disease. In Koch's postulates, there were 5 series of steps to follow in order to link a microorganism to a disease. The first would be to find an organism that is infected and has a disease. The second step in Koch's postulates would be to isolate a microorganism from the infected organism and attempt to grow it in pure culture in a laboratory. Pure culture would mean that only one microorganism were growing in the sample and not multiple microorganisms, which was very important for establishing a link between the microorganism, the single microorganism, and the disease. Step 3 would be to infect a healthy organism with the microbe culture. Step 4 would be to observe the organism to see if the same disease develops. And then step 5 would be to try to isolate the same microbe from the organism infected if the same disease symptoms do develop. Koch was very important in understanding disease causation and using Koch's postulates to do that. Lastly is the origins of life, which for several centuries were thought to be spontaneous, meaning that life could arise from non-living matter. This theory of life is called spontaneous generation, or abiogenesis. The first scientist to demonstrate that life cannot come from unliving things was Francisco Redi, who conducted an experiment with meat in 2 containers where he closed off one container with gauze so that flies could not come into contact with the meat. In the second container, it was open and flies were able to lay on the meat, hatch eggs, and those eggs hatched into maggots. Redi showed that in order to have maggots, you had to have a source of life, the flies, and if there was no source of life, maggots would not arise spontaneously from the meat. Supporting Francisco Redi's results was Louis Pasteur, who did an additional experiment to disprove the spontaneous generation theory of life. In this experiment, Pasteur made a swan-necked flask. He added a nutrient-rich broth to the flask, and he sterilized the broth by applying vigorous heat, so all life form in the broth was killed. He then let the swan-necked flask with nutrient-rich sterile broth sit on the lab bench for several days or weeks and concluded that no life was able to grow in the broth because life forms were not able to come into contact with the broth. The design of the flask prohibited life forms from getting past the bend in the neck, and therefore the broth remained sterile. So the conclusion from Pasteur's experiment, just like Redi's, was that spontaneous generation of life is not possible, and that in order to have life, you must have a source of life. So this new theory called biogenesis still holds today and says that life yields life and non-life cannot yield life.